Hey guys, yesterday we talked about setting the stage for sexual reproduction through meiosis, which is the cell division process that creates gametes or sex cells. And we just wanted to go back through those phases today. You should have a diagram that looks like this in your OneNote notebook, or you can download the PDF or draw it out yourself. It's totally up to you. But we're going to go through each of these phases today and just pick out the important things that happen, just like we did for our phases in mitosis. Now once again, we are looking at meiosis, and we want to make sure that we're starting on this cell with the homologous pairs right here. So the first thing that we have is prophase 1. In prophase 1, we have homologous pairs that are crossing over. So those homologous pairs, your chromosomes that are lined up right beside each other, they have the same genetic information in the same order, and so they're allowed to swap that information. So right here, homologous chromosomes or homologous pairs are crossing over. And once again, all crossing over is the swap of genetic information. We also have the nuclear membrane breaking apart, just like it does in mitosis. So our nuclear membrane is breaking apart. The next phase that we have in meiosis is metaphase 1. Now keep in mind that we remember that we have to put the 1 behind these phases because meiosis we go through PMAT 2 times. So this is the first time that we're going through PMAT in meiosis. And so we see that those homologous pairs that have already crossed over are lining up in the middle, and we still have our spindle fibers attaching to the centromere of those chromosomes. Our next phase is anaphase 1, and that's where our spindle fibers pull our homologous pairs apart. However, our sister chromatids are still together, so we still see our sister chromatids, which are the either side of the X of that chromosome that are identical. They have stayed together, but our homologous pairs that we saw over here that were together, they are now split apart. Our fourth phase in meiosis is telophase 1, and we see cytokinesis occurring right here, and cytokinesis is just splitting of the cell. We also see our nuclear membrane reforming. Our next phase is prophase 2, and in prophase 2, our nuclear membrane breaks apart again. Then when we look at metaphase 2, our chromosomes line up in the center of the cell, and our spindle fibers attach to the centromeres once again. Then we go to anaphase 2, where those spindle fibers rip the sister chromatids apart. Now remember, sister chromatids are just those chromosomes that have the identical X. And our last phase is telophase 2, where we have cytokinesis once again to create 4 cells, and our nuclear membrane reforms around the genetic information. And so what's happened is we have now made 4 different or genetically different cells that are going to be used in sexual reproduction later on, as we have made gametes or sex cells, and those examples are egg and sperm. There will also be a picture of my copy of this posted below this video, so you can kind of jot down some notes from that. You must make a submission of whatever notes you took, so whether you took them in OneNote and you can take a screenshot of that, or save it as a PDF or something like that, you can do that. If you wrote it on a separate sheet of paper, I just need a picture of that. Just make sure that you submit your notes in one way or another. Have a great day.